everyone. Welcome to this institutional seminar series. This particular seminar should have been last year in 2012, but because of several things that happened last year and even early this year, we could seminar given by scientists within ICRISAT until now. So Dr. Janila would be giving this seminar on groundnut breeding, rather groundnut improvement at ICRISAT using the genomic and genetic tools. Dr. Janila has been working with ICRISAT from 2011 onwards as a groundnut breeder. Previous to this, she was working at the Institute of Biotechnology at the Acharya Engineering Agriculture University, and before that in the Center for DNA Fingerprinting and Diagnostics for 3 years. And before that, soon after she finished her PhD from IRI in 2000, she worked at the Regional Agriculture Research Station of Angrao at Palem from 2001 to 2007. So here we are. We now request Dr. Janila to give her seminar, and it will be about 35 to 40 minutes, and then we will have time for interaction. Dr. Janila, please.
Thank you, Dr. Gautam. And good afternoon, everyone, and thank you for being here this afternoon. At the outset, I would like to thank Dr. Gautam for encouraging me to do this seminar, and also thank KSI unit for organizing this. And in this seminar this afternoon, I'll be talking about the genetic and genomic tools we use for development of new groundnut varieties. So, but before going into the topic, I would like to give a brief background about the global hunger index. As per FAO, every 1 in 8 persons, thank you. 1 in every 8 persons on this earth is hunger, which amounts to 870 million people. It's a huge number by any means. And from this pie chart, we can see this, the vast majority of them are concentrated in Asia and Africa. And FAO advocates that improving the productivity activities of these people can enhance the food availability and their income, and thus alleviate the burden of hunger, which means improving the agricultural productivity, because the majority of these people are engaged in agriculture and allied activities. So improving agriculture productivity will mean reducing the hunger in these countries of Asia and Africa. And incidentally, if you look at the global groundnut area, 96% of this 24 million hectares falls in Africa and Asia, which means that within agriculture, groundnut is an important crop in these two continents, and it's used for both oil and food purposes. 40% is used for food and 60% for extraction of oil globally, but then this percent can vary across in different countries. And it's an important cash crop for the reason that it has high value in the market, and the farmers can get a ready and very high income which they can use to buy their food. And most importantly, and which is dear to my heart, is it's very nutritious. It has very high oil content, about 48%, and hence energy rich, and about more than 20% of protein, and it's rich in micronutrients. So it can be very vital in fighting protein, energy, and micronutrient malnutrition in these countries. And this is a screenshot I have picked up from this company NutriSet, which makes plumpy nut. It's a groundnut based product which is patented, of course, which UNESCO uses extensively in its acute malnourishment eradication programs in several countries in Africa and Asia. It is a ready to use therapeutic product which this company makes and sells to UNESCO. And I mean, with this background, I would like to say that groundnut is an important cash crop, food crop, and then it improving productivity improves the livelihoods and then reduces the hunger of millions of people across Asia and Africa. And it is for this reason that ICRISAT initiated the groundnut improvement program in 1976. And when we talk about groundnut improvement as a whole, improvement of and development of new varieties constitute an important part of the solution. And of course, it's not the only one, as I will be discussing. With this background, I have structured my presentation to briefly discuss about the basis with which, or based on which we define our objectives in breeding, and then large majority of my presentation will talk about various tools. I'll not be making an example listing of about each of these in detail, but then I'll bring about examples wherein we actually use them in our current breeding programs, and then about IMO developmental pathway, which of course is our strategic backbone kind of a thing as we do all of our activities. And then coming to challenges, I'll not again discuss elaborately about all the challenges, but then more specifically about the nutrition and aflatoxin. And then in a couple of slides, I would summarize my presentation. So coming to the basis, it is the biotic and abiotic stresses and the challenges across the production environments which define largely our breeding objectives, which are dynamic and ever challenging, and they become even more challenging in the light of the climate change aspects. And next are the preferences of all the people involved along the value chain of it, from the producer till the consumer. And nutrition, and of course IMOD strategy and the gender, these are all they all form the basis as we define our breeding objectives. And of course the yield gap, not the least, it in fact overlaps with other several things. If you look at the yield gap of groundnut, we can see that in Africa, where this yellow and yellowish green spots are there, means productivity yields less than 1 ton per hectare, and these green spots, which indicate about yield about 1 to 1.5 tons, which means to say that there is huge yield variation across the growing regions, and this variation is very high, 300 to 5,600, which itself means there is an enormous potential to increase the productivity levels in these yellow and green regions to make them blue. And hence plugging the yield gap is an important breeding objective. And this is this graph I have picked up from an old study which was conducted here at ICRISAT in 1990s, which is relevant even now, where the realized yields in the farmers field are hover around 1 ton, while the potential which is possible is as high as 8 tons per hectare. And even if we are coming close by somewhere midway, even that would be a huge leap in terms of yield and productivity. And how do we plug this? So it's not by one means, but through several of these things coming into picture, and improved varieties are one of them. And here is the toolkit which I would be discussing under 6 different subtopics. So the first one to begin with is a genetic variability, which is vital. It's like vital oxygen. Oxygen, as oxygen is for human being, it's like genetic variability is like that for a groundnut improvement person. And we have to live with the fact that the available variability in the groundnut is limited because of the fact that it has evolved from a single hybridization event followed by chromosome doubling. And then we could not, the cultivated groundnut could not get good genes from its wild species because its haploidy levels are different. Although there are 81 species of arachis, only Arachis hypogaea and Monticola are tetraploids, rest all are diploids. So there are crossing barriers imposed on it. And then the induced variability, that is through mutations, both physical and chemical, the success of which I'll talk a little later in the next slide. And attempts were made by different groups to utilize these wild species through specific hybridization. And there are several derivatives, and we have good success stories, particularly with regards to foliar disease resistance, although we have to bear the burden of linkage drag in terms of reduced quality of pods and the kernels. And then the gene banks and the ongoing breeding programs across various institutes are the repositories of huge sources of variation. which the breeder can rely upon. And having known about the genetic variability and the source of variation, the next important aspect comes about the understanding the genetics of it, because that defines how the breeding strategy could be. I'll mention here about our most recent work using interspecific derivatives for late leaf spot resistance. This is one of the important biotic challenge we face in Asia. And the study has indicated that the resistance is governed by both maternal and nuclear genes, and field measured traits are good enough because they have high heritability. What does this say? This uh, usually in our breeding programs we use the donor as a male parent, and this kind of insights into the genetics gives us a clue that no, they can be used as female parents to tap the maternal effects of them. So genetics and trait association similarly give us the way we go about with improving a particular trait, and also sometimes the ease of our work. For example, here we have water use efficiency. Although we all know that this is an important trait, but then measuring it could be very difficult and rather not feasible for a breeder to do regularly in the breeding activities. But then the studies have shown the close association of water use efficiency with the other two traits, which are rather easy to measure and they're robust in nature. And coming to the breeding methods, introduction mass selection, of course, are the ones which were used initially when the breeding programs have newly started. And of course, mutations also, they have contributed enormously. And there is a report from China which says that 50% of this is the record by 1996, 50% of the varieties are from mutations. And they cover an area of 19% of groundnut cultivated area at that point of the year, which means to say that their contribution has been quite very significant. But the most commonly used breeding method is hybridization and selection. And here in hybridization, we use two or more parents to bring the genes from these two. Like I have a schematic picture here wherein we have pooled all colored beads together. And then through the process of selection and the way we handle the breeding material, we string them in the fashion we want, in the particular pattern we want. The fashion and pattern, which I mean that the type of plant we want, a drought tolerant, a disease resistant, high yielding, high oil content, all these things. So, and these are the ways we handle the breeding material. Of course, the most common ones are bulk pedigree and single seed descent methods, although with the use of genomic tools, Bacros has coming into the picture more lately. And here I want to, although I have just summarized the entire breeding method in one slide, I want to mention here the untiring efforts of the technical staff of groundnut breeding, because success rate in when you make crosses is very low. And when I've talked with several of our colleagues at different breeding institutes, I realized that the success of crossing percentage which we get in ICRISAT is enormously high by any means, thanks to all of them. And this is our hybridization block in one of the seasons. And coming to the selection again, I have described about the ways we handle the breeding material, and then coming to the screens, phenotyping tools act as the screens by which we reject the unwanted ones and pick up the ones which we want to forward them. And I have mentioned screening tools for three of the traits over here. And the first is about the foliar diseases, which are very important because these diseases alone cause losses as high as 30% to the total yield. And we have both the techniques, that is field screening and the detached leaf technique. In this field screening, you can see here planting of an infected plant and then spraying of the spores. And here we can clearly see the field, the infector rows already damaged by the disease, while here are the entries which we are screening for them still green and all. And similarly is here the detached leaf technique, wherein we put the leaf on a sand and then screen for disease. And coming to the phenotyping tools for drought, here at ICRISAT, we use a combination of both empirical and trait-based approach, because it's been known that both these contribute, uh, give equal results, but then we are using a combination of these two. Here is the empirical approach wherein the drought stress is imposed, and then we look at the yield produced by a particular genotype under stress conditions and select such genotypes where the yield reduction is not very high, even under imposed drought conditions. And here you see the trait-based approach which we do for SCMR and specific leaf area measurements. And when we are talking about phenotyping tools for breeding, they have to be robust, which means that a large number of samples in a fairly short amount of time, they have to be completed. And here we have this NIRS equipment for which we have acquired to determine the oil content and protein content and fatty acid content of our breeding material, as improving for oil content and altering the proportion of fatty acids is one of our breeding objectives to enhance the content and oil quality. And here comes the cars which Rajiv often mentions. And before going into them, how we are using them, let me indicate that neem term is the first variety which has been released in groundnut using marker-assisted breeding approach from USA, which is nematode resistant. And this was followed by another variety which is both nematode resistant and also has high oil quality. And then we have to, at this stage, we have to accept the fact that when contrasting the adoption of these technologies in various developing and developed countries, it's happening at a very slow pace and rather not happening in some of the countries in Asia and Africa, while it's happening at a much faster pace in developed countries. While on the other side, the new tools like genomic selection tools and all that are fast emerging. Given that background, at ICRISAT, we are using these new tools for improving resistance for foliar fungal diseases and also for improving oil quality. And we are at fairly advanced stage for breeding for rust resistance, wherein we try to introduce a major QTL which has been identified and known to contribute more than 80% of the phenotypic variation into three popular varieties in India, that is ICGV 9114, JR24 and TAG24. So here you can clearly see this is the recurrent parent and this is a donor parent GPBD4, and this is an introgression line at an advanced stage which is clean from the disease. I'll show the disease course in the next slide, and this is the field wherein you have a susceptible recurrent parent donor, and these are the introgression lines on both sides of these parents. And if you look at the disease score, GPBD4 has a score of 1 and which went up to 3 towards harvest, while the susceptible ones started with 2 and 3 at 75 days after planting and went up to 9. And the introgression lines, which we have screened in the last rainy season in a replicated plot, have shown at least, we have screened over 200 of them, but 77 of them at least had a score of 2. And these will be going to the yield trial the next season. That's a replicated yield trial. And then, as I mentioned, this MABC program is also on for improving oil quality by introducing two mutant alleles, which are known to improve the O by L ratio of the oil. And then the program is also on because now we have another QTL from the same donor for late leaf spot resistance also. So gene pyramiding for these both resistance for both these diseases is also on. Here we use different recurrent parents like having high oil content and the one which is drought tolerant, so that we are looking at a multiple trait breeding, not traits in silos. And the most important, although I have put it last, is the participatory breeding. And I hope you all agree that this is very important because, particularly for crops like groundnut, where there are huge issues of seed systems, this paves way for wherein the farmers start owning and then adopting those new varieties. And through this approach, under TL2 program, we could identify and release several varieties across Asia and Africa. And the strategy of IMO developmental pathway is particularly important for groundnut for the reason that all along the value chain, you talk about subsistence or high value agriculture, which is oriented to export markets, the groundnut, all the stakeholders involved in groundnut production are involved in this. So this strategy becomes even more relevant, and I have two cases to document or to say as examples for this. When we have high oil lines developed at ICRISAT, and when we are looking at the commercialization of these lines, we would need to answer several questions related to markets and seed viability. Markets, why? Because if we have a high oil line, would it trickle down to increase income to the farmer or not is a big question. And then a study conducted at ICRISAT way back in 1985 has shown that it trickles down to the benefits of the millers, trickles down to the farmers in the form of increased income. So high oil lines can contribute to increasing income of the subsistence farmers, which are at the lower end of our IMO developmental pathway. And then seed viability is another question, the science-based question probably. So already we all know that seed containing oil content loses viability fast because of the oxidation, and further increasing the oil content would it hasten the process. So we have conducted a study And we have shown that there is no such association. The correlation is very weak, as we have shown it here with increase in oil content and seed viability. That was very interesting finding. And it's like a green signal for commercialization of these high oil genotypes in the farmer's field. And another aspect of orienting the subsistence farmers to markets comes the dual purpose genotypes. A study conducted by ILRI has shown that an improved variety in Anantapur has resulted, could contribute to increase in milk yield of milk cattle up to an 11%, up to 11%. So such studies and have been documented. Studies have also been documented that there is enough genetic variability which can be tapped, and the good thing is there is no inverse relation. So there are no trade-offs with the pod yield, which means that we can improve the pod yield and at the same time have good yield and quality, not compromising one for other. And moreover, they support, they not only give system sustainability to the systems, because they also increase the income of the farmers, because they can be engaged in another allied activity, not just being in the production of groundnuts. And they are a good they complement nicely with the cereal fodder because they are rich in protein and minerals. And coming to the addressing challenges, I said that there are several challenges along the way. Right from the development of the product, like the development of the products, we have challenges in defining the when we define the objectives, we have challenges and new challenges coming up. Like we talk about the climate change effects, now we are working on heat stress. I mean, varieties tolerant to heat also, in addition to the drought. And these are the several challenges. But as I said, that I would be discussing about nutrition and aflatoxin in this section. Nutrition, uh, we all know that groundnuts are very nutritious and they can contribute enormously to enhance the nutrition of the farm families. And we know that, and I've seen that in Bihar and elsewhere, and I also picked up information from Africa and several other countries, that groundnut is cultivated for household consumption or to supply for the local markets in very small patches. Okay, and the seed which they get for those patches is the one which is available with the shopkeeper. They buy a kilo of that seed and then come sow, and they use it for their either home consumption or for the just selling for a day or two in the local market. But that's very critical for their own nutrition. So how to address this? So we would need some new ideas and things like that when we look at this, because I still see, although it's very small area, I still see a potential of it in enhancing the nutrition and also engaging the women in these activities. And another important thing is aflatoxin. So I'll not go about describing what is importance and all that, because we all know about it. But we have to live with the fact that we don't have sources of resistance for this in the available germplasm. So probably for such kind of problems, we need to rely on new technologies like transgenic technologies, which already are underway at ICRISAT. And these are the two things which I thought that I will discuss with regards to the challenges, although there are several of them. And then would like to summarize my presentation, saying that conventional breeding procedures along with phenotypic tools, when I say conventional breeding procedures, it also includes the participatory breeding approaches, have contributed to development and release of a large number of varieties across several countries in Asia and Africa. And these are the numbers. So far, since 1986, 138 such varieties have been released in 36 different countries by the national partners there, with whom we work in close partnership. And this is an example which has happened very close to ICRISAT in a district called Anantapur, which has the largest groundnut growing area in the world, about 800,000 hectares, where a variety has been introduced through participative breeding approach and was identified and released in 2006. And then since then being commercialized in that. But I want to end saying that new improved varieties are an important part of the solution, but they alone cannot contribute to the developmental impacts which we envisage. So for developmental impacts to be achieved, we would need ICM practices going along with the improved seed and being adopted by the farmers, and seed systems, particularly for groundnut, which are very crucial and important given the volume of the bulky nature of the seed and the amount of seed raised we would require. Partnerships both for upstream research and the downstream developmental activities, uh, for technology delivery and seed systems and several of these things, and and several of these things which I have listed, including the favorable policies. I have learned about the policy which could be quite encouraging for groundnut, that varieties which are older than 10 years will not get government subsidy, which means that I'm hopeful that at least now the TMV2, which has been ruling for over 7 decades in Andhra Pradesh and Karnataka, which have a large area under groundnut, would now fade away given these policies. And more such policies and favorable environment to take these newer technologies to the farmers could be needed in this place. With that, I would like to acknowledge our groundnut team members, because the work which I presented is not done alone by me, but the entire team is involved with this, and the team guided by Dr. Gowda. And I also acknowledge groundnut farmers, partners, and tree research, and of course the consortium. And with that, I would like to say thank you.
Yeah, Janila, just to comment on the surrogate you were talking about for water use efficiency. Well, that was maybe what was used 10 years ago. We have sort of tested that. Forget about SLA, forget about SCMR. There are really no good sort of surrogate. We need to really sort of look forward and move forward there. My question is, what would be your approach to tackle the extreme variability of drought situation? So in other words, are you going to be trying to develop a one-fit-all type of variety, or do you think there is a need to look at specific environment and with specific type of stress to better target sort of breeding efforts?
I would look at specific type of environment and stress, and then think about what kind of variety would be suitable for that. First, we define it properly. It's not that drought tolerant across Asia and Africa. We have a specific environment in mind, and then we look at what exactly is happening in that stress environment, and then think about like what could we see, envisage to see in the variety to suit that particular stress. So it's for the end of season drought kind of a situation wherein we look for short duration as a mechanism of escape. So just to escape that. Yeah, like several things come into play when we talk about end of season, like aflatoxin contamination, enhancing.
Janila, the topic was very global and general, but yes, that may be the reason that you have not discussed the regions, global regions. That so still I'm looking for the information, and you can answer now also. That what are the major challenges the crop is facing in major regions where this crop is grown? And we know these tools, but how the crop groups are addressing those challenges? So can you prioritize the challenges in this crop in different regions, and what are the basically how crop groups are handling those challenges?
I mean, I thought that I made this presentation more specifically, not globally, because although we know that these challenges are, several of them are global, and then some regional specific things would come into picture. But then while I was going through all along, I was talking about these challenges which we at this center are addressing. For example, I talked about foliar diseases, I talked about drought resistance, I talked about oil content in the seeds, because particularly for South Asia, that's very important because 80% is crushed in India. So, and then I also talked about improving the oil quality also and other quality aspects of it, reducing the nutrition. So then that's one thing. I thought that I made my presentation that way, not looking from a larger picture, but then being more specific on what work actually we do over here. And then, as you know, that groundnut breeding program, we have at ICRISAT, we have 3 regions: Eastern and Southern Africa, and WCA, and South Asia. And for South Asia, we have 3 different groups, grounded groups, working, addressing these local challenges at each of these regions. And we, being at, we place at South Asia, look at most common problems of South Asia. For example, if you take about the biotic stresses, it comes with diseases. And of course, viruses too also in the localized species, although it's not a major thing. It's more important in Africa. The rosette disease, virus disease, is important in Africa, not in Asia, which our counterparts in those countries are working. And drought is common across all the regions, but then, as Dr. Gowda has just now mentioned, the time of drought and the kind of stress, and in some places it's a combination of drought and heat stroke, and at some places it's not so. And in some places, like when you move towards east of Asia, the problem of cold tolerance is there. I haven't touched more about it because that's still not as seen as a very major problem, although it's for China, and we don't work much for them, of course. And this is regarding the drought and all. Coming to the consumer preferences, there are 2 major uses: food and oil uses. That's what I tried to mention. And wherein in South Asia, it's mostly, particularly when you come to India, it's mostly oil focused. That's the reason why we are trying to enhance the oil content in the seed, so that the benefits trickle down all along the value chain. And then food uses are also there, and they're rather increasing, and hence our concentration has also been on those to increase the size and such kind of things. Of course, I didn't talk much about it, just made a passing statement on oil quality, wherein I mentioned that we use robust stones. Raju, given a choice, the breeder would always be dreaming about breeding for multiple traits together. But given the tools we have, the existing phenotypic tools and all these things, even the genomic tools as of now which we have, that does not allow us. To a maximum extent, will be able to pyramid one or two genes. But then, when the tools are available, like the genomic selection tool, wherein we would be able to focus on several traits at one go, definitely the breeder would love to use them. Even now, I would say that breeder's target is never a trait in silos. When we are trying to improve disease resistance, we try to use a recurrent parent which is like suitable for the particular environment, like it's drought tolerant, already has some good features in it, its quality is good, or its pot shape is good. So already certain characters are taken into it, and then we try to add on one or two which are not there with the current available tools. So that's one thing. So multiple traits, yes, that would be the most preferred one, given the tool to do that. I mean, we are lacking in that. And coming to the second question on my take on this aflatoxin thing. So this aflatoxin contamination can be tackled by two mechanisms. One is host resistance, and a large part of it comes from the management. So while we'll not discuss most of the management thing over here, coming to the host resistance, as I said that we are limited, constrained by the available sources of resistance in the available germ plasm. And hence comes the transgenic technology. And I think I'm not competent enough to say which gene exactly to be used or which approach with regards to transgenic. But then whichever works, like we have examples in other crops, like we say about BT cotton, it works fantastic. If something works like that fantastically, it's good.
Janila, I can. Yeah, Groundnut is a fantastic biological nitrogen fixer. Nevertheless, it faces a constraint in certain area. It's not part of PL4. What is your view on that? I mean, is there not a huge potential of tapping on? I think a great genetic variation. You got a paper with Michael a year ago where we look at the absolute absence of trade-off between full content in nitrogen and body yield, which means that you can have material that produce huge amount of nitrogen, all fixed or most of it fixed. So is there not a potential, thinking more, West Africa, loamy soil, absolutely devoid of nitrogen, extremely sandy. Is there not a huge potential there?
Yeah, I agree with you, and I feel that Groundnut shouldn't have been dropped from that product line, rather it should have been improved. But that's a fact that it's been dropped now. But I agree with you, and then I feel that there is a potential for that. Working on in that area in Groundnut. But then what I gathered, probably I should say from my predecessors, quite a good amount of work has gone into it, and they have seen the genotypic huge genotypic differences at the host genotype and also at the bacterium genotype, and there were huge possibilities to improve both of them together simultaneously, and then double the effects of nitrogen fixation and all these things. But then probably the decisions made at ICRISAT at that time, it's like more of an NRM issue wherein that particular microbiome or whatever it is has to be added to the soil, and also the research has been put to back burner at some point of time, and probably never pursued. Although given a chance, I would like to work with this area.
This is a comment and doesn't require any response from Janila, but just personally raised to certain issues. In case of aflatoxin, though we say that we don't have anything good in cultivated type, but the fact remains that very few, maybe not more than 2,000 times overall, only 1,500 is available. But considering the level of resistance we have, that kind of level of resistance is higher than background has already been taken. And a line companion of 99114, 89104 was in fact aflatoxin resistant line, was doing equally well in 114, and out of these two, one of them was selected. aflatoxin that kills microbes. The fact remains that phenotype, accurate phenotype for aflatoxin is a problem, unlike rust resistance, where if you go in rainy season, or like that, anybody can select for rust resistance. So in fact, genomic tools for traits, not complex traits, that's a trait may be still simple, but because they are difficult to phenotype, those traits we should have. And in fact, my preference, beside continuity, if we can add something transgenic which can block synthesis of aflatoxin, either in the cultivars or seed infection in the field or in vitro seed colonization. That would be that has to be understood. But aflatoxin, yes, it's a problem. Genetic resistance available in few hundred lines has been evaluated, has been more. Wild you find, but then those wilds are such difficult that you have to really use a stone to break those bonds. There's a physical barrier which cannot be used in a cultivated tank. Our attention should be, in fact, aflatoxin for transgenic as well as for using the multiple methods.
which we are using.
Because you do 3 replications. In 4 replications, will be so high. In 1 replication, it will be 0. And you can't believe either of them.
But
It's not that easy.
Yeah. I'm just thinking that leads to another question, whether genetics is playing any role there, or just assuming.
See, again, once we have the product line coordinators, I think this is one thing that we need to do, not only for but even for other areas, including drought tolerance, looking at what Vincent was mentioning, what kind of drought tolerance that we need to be looking at, whether the surrogate traits that we were using earlier, are they really the traits that we should be looking at, or should we be looking at something else? So those are the things that we need to look at. So before I close, Janila, because this was, at least for me, it was the first seminar that I heard, and just a few comments that I would like to make so that you can enhance the future presentations. One is that among the total number of slides that you had, only 1/3 were on the real groundnut work. So the others were, auxiliary. So we need to enhance maybe 2/3 on the subject and the others introductory and conclusion. You mentioned, you put one slide of hunger map, and you said groundnut is helping in reduction of hunger. I think looking at the protein content and oil, I think groundnut is more of trying to address malnutrition rather than reducing hunger. I think because hunger is other crops like cereals and other things. So that is. Yeah, you can, as I said, these are only comments as I understood. The other thing that I wanted to mention was you said lacrosse we are using more because MABC has come. I think lacrosse has been there, Alpha as well, has been there. They're only using markers in selections. So lacrosse has been there, Alpha has been there. So when we make comments, things need to be careful how we make statements. In terms of yield, you mentioned 300 kilograms variation, 5,600. But there are reports where people have got more than 10 tons. In Zimbabwe, golden days, there used to be a club called the 10-ton club of farmers who would consistently get 10 tons per hectare. So when we say a range, it should be for whatever it is, 300 or 500, more than 10 tons. So that is the potential is there. Of course, the 10-ton variety was possibly 160 or 170 days. Doesn't matter, but it was there. So I think those are a few facts that we need to be cautious when.
Can I answer that?
I said, yeah. These are only comments.
Yeah, no, but then I need to clarify them. Probably that was not meant along correctly. The first thing is regarding the global hunger map, which I showed. And then I said that yes, the groundnut can be not only malnutrition, it can fight hunger. That I want to reiterate again, saying that it's an high value cash crop and the farmer can sell the produce in the market, and with that income he can buy food for his family. So it plays an important role because of its value, and I don't agree that it's only the cereal which can do for hunger. That's one thing. And coming, no, I say that I'm contributing. Okay. Given the fact that it gives high income. And the second thing is backcross. I said that it's a breeding program. Always, has a long history of it, and we have been using. But then I said that it gained more prominence with the marker extra backcross because we started using more. So earlier it was only pedigree, bulk pedigree, and single seed breeding method which we were using, and less of backcrossing method, less of compared to the other three methods. That's what I mentioned. That's what I said. That's what I said. Yeah. So that little bit of prominence also it got because of this marker arrested back crossing thing. It's just limited to that. That's what. Okay. Okay. No, yeah, sorry. And this groundnut yield variation also, this is average of different countries. 300, there are countries having an average of 300 and 5,600, and not the actual yield potential. I mentioned about a yield potential in another graph wherein it's as high as 8 tons per hectare and also 10 tons per hectare. Yeah. The real, that's a study conducted in ICRISAT. So based on this 1990s, I said that's a study which is as good as. Yes, I know there are isolated cases of it. I agree.